Hello students, welcome on board. In this video, you will be learning few topics from the chapter Sexual Reproduction in Flowering Plants. This is part 1, wherein you will be learning about the male reproductive unit, the process of microsporogenesis, and the structure of a pollen grain. Without further ado, let's begin with the first topic. Now, first we need to know that the flowers are the site of reproduction in flowering plants. Flowers are the site of reproduction in flowering plants. So a flower will have a female reproductive unit and a male reproductive unit, right? Now in this session, we'll be learning about the male reproductive unit. Male reproductive unit. Now the male reproductive unit in a flower is called the stamen. It is called the stamen, and it is also known as androecium. Is also known as androecium. So the male reproductive unit of a flower is called stamen, and it is also known as androecium. Now the stamen basically has two parts, an anther and a filament. So this is the anther, and the stalk is called the filament. Now the anther is said to be bilobed and diathecous. Now if you look at the anther, there are two lobes, right? Because it has two lobes, it is said to be bilobed. Now imagine taking a lobe and cutting it open. So what will you have? You will have two cavities, right? So these two cavities are called thecas. Because there are two thecas, it is called diathecous. So the anther is said to be bilobed and diathecous. All right. Now let's see what an anther looks like. Now here you can see that the anther has four sides, right? These are the four sides. These are the four sides, and within the anther there are microsporangia. These are microsporangia. So we can say that the anther is a four-sided structure. It is a four-sided structure consisting of microsporangia. Anther is a four-sided structure consisting of microsporangia. These microsporangia then later produce pollen grains, which we'll be learning shortly. Now let us learn about the layers around microsporangia. So the microsporangia basically has four layers, and these layers are epidermis, endothecium, middle layer, and tapetum. Okay. Now the first three layers, that is the epidermis, endothecium, and the middle layer, are protective in function. And the final layer, which is tapetum, provides nourishment to the developing pollen grains. So the first three layers are protective in nature, and the tapetum provides nourishment to the pollen grains. It provides nourishment to the pollen grains. Now we are going to learn about the process of microsporogenesis. Microsporogenesis. All right. So we've learned that the anther consists of microsporangia, right? Now at the center of each microsporangia, there is a tissue which is called the sporogenous tissue. It is called the sporogenous tissue. Sporogenous tissue. Now when the anther develops, the sporogenous tissue undergoes meiosis to form microspores. Okay. Now, these microspores are arranged in a cluster of 4 cells, which are called microspore tetrad. All right, it is arranged in a cluster of 4 cells. Now, when the anther matures and dehydrates, when the anther matures and dehydrates, due to the loss of water, these microspores dissociate from each other to form the pollen grains. All right. Now, because the sporogenous tissue is the mother of microspore and pollen grains, it is also known as microspore mother cell, microspore mother cell, mother cell, or pollen mother cell, pollen mother cell. So, the process of formation of pollen grains from the sporogenous tissue through the meiotic division or meiosis is called microsporogenesis. Now, when the pollen grains are produced, these are released by the dehiscence of anther, by the dehiscence of anther, which means that the pollen grains fall off from the anther. So, this is the process of microsporogenesis. All right, we just learned about the formation of a pollen grain. Now, let us see the structure of a pollen grain. All right. So, a pollen grain represents the male gametophyte of a flower. It represents the male gametophyte of a flower. All right. And the pollen grains are usually spherical in structure. And they have two layers. The outer layer is called exine and the inner layer is called intine. All right. Now the exine is made up of a substance called sporopollenin and intine is made up of substances called cellulose and pectin. Now if you observe the outer layer, you can see a depression here. This is a region where the sporopollenin is absent and this region is called germ pore. And within the pollen grain, there are two cells, a large cell which is called vegetative cell and a small cell which is called a generative cell. Now the vegetative cell contains food reserves and it provides nourishment to the male gametes that are produced by the generative cell. So we've learned that the pollen grains have two layers. The outer layer is called exine and the inner layer is called intine. Exine is made up of sporopollenin and it is a highly resistant substance. That means no enzyme can degrade this layer. And intine is made up of cellulose and pectin. Now the region where sporopollenin is absent is called germ pore. Germ pore is the region where the sporopollenin is absent, right? We also learned that there are two cells within the pollen grain. The large one is the vegetative cell, which contains food reserves, and the smaller cell is called the generative cell that produces two male gametes. And these male gametes obtain nourishment from the food reserves stored in the vegetative cell. All right. So that's it for this session. This was part 1 where we've learned about the male reproductive unit, the process of microsporogenesis and structure of a pollen grain. In part 2, we'll be covering female reproductive unit, Megasporogenesis and Embryo Sac. If you found this helpful, please do like the video. And if you have any doubts, please comment down below. Subscribe to watch the upcoming videos. Thank you for watching.